Hello, my name is Josh Mahlios. I'm a Senior Marketing Product Manager at Miltenyi Biotec, and thank you for joining this webinar presentation about the rise of neuroimmunology and about the tools and solutions that we offer to help you navigate the research and translational challenges during the next revolution in human health. In today's webinar, we will cover the following topics, starting with the history of Miltenyi Biotec, followed by a brief background of neuroimmunology. We will then take a look at the different products and technologies that together comprise the complete workflow from Miltenyi Biotec. We start with the importance of sample preparation, followed by the core of MACS technology, which is magnetic cell separation. Next, we will discuss which factors to consider for reliable and reproducible cell culture and phenotypic analysis of target cells. We complete the workflow with our new imaging platform, and will then move on to some example publications for various neuroimmunology-related applications. Finally, we will wrap up the webinar with a quick summary and supporting materials. We will first start with a brief introduction of Miltenyi Biotec. Miltenyi Biotec was founded in 1989 by Stefan Miltenyi, and the basis of MACS cell isolation is the column technology developed as a part of his diploma thesis, with the technology being patented in 1990. The potential for MACS technology to become a revolutionizer in the field of gene therapy became obvious, and the gentleMACS CD34 reagent system for selecting hematopoietic stem cells from donor apheresis was developed. The clinical evolution of the company has continued for over 20 years, including setting up a GMP manufacturing site in Tetterow, Germany, and in 2014, the FDA gave approval for CD34 isolation using the gentleMACS together with the gentleMACS CD34 reagent. What started out as a diploma thesis has since grown into a global company of 3,000 employees and over 20,000 scientific publications citing MACS technology. Annually, more than 2,500 patients receive cell products manufactured with Miltenyi Biotec technologies. While we have thousands of products, instruments, and services, as a company, we have one mission. The words empowering discovery and advancing therapy define our company's mission, which is to improve scientific understanding and medical progress by providing solutions that advance biomedical research and cellular therapies. As a company, we are able to achieve this mission by our ongoing commitment of bringing newer and smarter technologies to the scientists and clinicians who are paving the way to longer and healthier lives. So why are we here talking about neuroimmunology? Let's take a look at a little bit of the history and rationale for focusing on this topic. Neuroimmunology is the interaction of the immune and nervous systems, two of the most complex biological systems known in nature, with significant scientific and financial efforts directed towards the development of approaches to diagnose, treat, or prevent neurological diseases. For decades, the brain was thought to have immune privilege, operating autonomously from the immune system. This has been systematically disproven by numerous studies showing the extensive interactions that exist between these two biological systems. Neurological disorders with immune components have been documented for centuries, though the etiology and treatment of many of these neurological disorders remains elusive. The recorded history of neuroimmunological disorders dates back over 400 years, with the first recognition and documentation of such conditions such as multiple sclerosis and ALS. Additional conditions were later identified in the 19th and early 20th centuries, and these include Parkinson's, Huntington's, and Alzheimer's disease. Fast forward to the 21st century, and there have been numerous advancements in our understanding of the disease pathogenesis, and many failed clinical trials have been held to combat these various diseases. These trials include failed vaccines for Alzheimer's disease and attempted stem cell therapies and targeted depletion of B cells for multiple sclerosis. So if neuroimmunology has been around for hundreds of years, why is this webinar about the rise of neuroimmunology? To understand this rationale, we need to consider the current and future threats to human health, and how the relationship between the brain and the immune system is likely to be at the forefront of human health in the future. When we look at how the top 10 global causes of death have changed during the 21st century, we can see that diabetes, heart disease, and Alzheimer's disease have become a more common cause of death. But what about the future? Let's take a look at what the World Health Organization thinks the trends are going to be, focusing on the top 3 causes of death, being heart disease, cancer, and neurological diseases. When we look at the predicted cause of death over the next 40 years, we can see that brain diseases are anticipated to become more and more of a health threat, especially in wealthy nations. In the U.S., cancer and heart disease are the leading causes of death, but we are seeing a slight decline in the prevalence, with neurological and infectious diseases making the greatest gains. What we see and perceive to seem to be different things when it comes to death. This graph shows what we actually die from on the left, what we search for on Google, and what we see on TV, all of which are quite different. This is demonstrated in this graph, depicting on how under or overrepresented causes of death are reported by the media. NIH funding, however, obviously has neuroscience as a priority, as is evident by the projected funding for 2020. There are a number of different immunological example applications. It's broken down into 4 primary categories, the first being cancer, with the most prevalent being glioblastomas. There's also astrocytomas and adenomas. There are immune neurological disorders such as multiple sclerosis, SLE, or CNS vasculitis. Degenerative diseases include Alzheimer's disease, Parkinson's disease, ALS, and Huntington's disease. And the other category includes stroke and traumatic spinal injury, infection, psychiatric conditions, and genetic conditions, all of which have been found to have immunological components. Now that we've gone over the history of neuroimmunology a little bit, now we're going to focus on the workflow from Miltenyi Biotec. This goes from sample preparation to cell analysis. So we start with a solid tissue, and we move on to our sample preparation. This is followed by cell separation and various downstream applications such as cell culture, cell analysis, single-cell sequencing, or proteomics, and we're also introducing our new imaging platform. We're first going to cover the importance of sample preparation. So this section will cover sample preparation, one of the most critical aspects of any workflow. The quality of your starting material will define the quality of your results. The basic principle of sample preparation of taking a solid piece of tissue and using both mechanical disruption and enzymatic digestion to dissociate the tissue into a single-cell suspension. There are 3 primary goals that need to be achieved to reproducibly generate single-cell suspensions of viable and recognizable populations. Whether brain tissue, tumors, or organs, you begin with a mixture of cells embedded in the extracellular matrices, basal membranes, vasculature, fat, and possibly necrotic tissue, often making target cells difficult to isolate and characterize. The first goal is to consistently dissociate your starting materials through a combination of enzymatic and mechanical disruption. Common methods include generating concoctions of enzymes from various vendors that widely vary in source, activity, purity, and even nomenclature, and combining this homebrew with a manual dissociation process using razors, scissors, possibly mashing, pushing tissue through a mesh or bead mills. The result is a dissociation process that is difficult to reproduce due to the number of product and individual variables. The second goal is to preserve cell surface epitopes, which are essential for the identification and characterization of various cell types and subpopulations. In addition, preserving cell surface epitopes may be essential for downstream applications and functional analyses. But how are you to know which epitopes are susceptible to enzymatic digestion, and at which concentrations, and for what duration? Not knowing which epitopes are affected by your sample preparation protocol can lead to misidentification of subpopulation and disruptive artifacts. Finally, maintaining cell viability and functionality is essential for any meaningful downstream analysis, so minimizing the amount of both the mechanical and enzymatic dissociation, along with the utilization of consistent and defined reagents, is paramount for success. We know that these goals are important, but consistently acquiring defined reagents in combination with human variation can make this process very labor-intensive and variable. Fortunately, we have a solution in the gentleMACS. With the gentleMACS octo-dissociator with heaters, we offer a fully automated tissue processing instrument to standardize the dissociation and homogenization of your starting materials. With this technology, we combine mechanical dissociation and enzymatic digestion on the same instrument. 8 samples can be processed at once, in parallel or independently, and every position is individually tempered for efficient enzymatic dissociation. Here, different applications can run on different positions at the same time, or you can start experiments on available positions, even if Others are occupied by other lab members, making it a great instrument for diverse laboratories. We offer a wide range of protocols and kits for the applications of cancer, neuroscience, immunology, stem cell, and cardiovascular research. It would encourage you to visit our website to discover the breadth of our technologies and supporting materials for your research. The next step in the neuroimmunology workflow is cell separation, which takes us to the next section that will describe the fundamentals and advantages of MACS technology. In this next section, we're going to be talking about cell separation using MACS technology. MACS technology is based on three principles. First, magnetic labeling, where an antibody conjugated to a tiny paramagnetic microbead binds to a receptor or protein on the cell surface. There are different strategies for this step, which will be covered in more detail later in the presentation. Second, we have magnetic separation, the process where cells are passed through a column that is positioned within a magnet. The cells labeled with the microbeads are retained in the column, whereas everything else passes through in a gentle way. The flow-through is called the negative fraction containing the cells without the antibody. Third, in the elution step, the column is removed from the magnet, enabling the paramagnetically labeled cells, meaning that they lose their magneticism outside of the separator, which allows them to pass freely through the column. This can be achieved through a quick manual step using a plunger to gently elute the cells, or via an equally gentle automated process using a vacuum to pull the cells out, or increasing the flow speed of the buffer in the system. All three methods are gentle for the cells and separate the cells into a negative and positive fraction with exceptional purity and high viability. These key components are the MACS microbead, the MACS column, and the MACS magnetic separator. There are several versions of each available, depending on your starting material, cell type of interest, and whether you're looking for a manual or automated system. Since the column is the core of MACS technology, let's take a quick look at the benefits and why it is clearly the superior choice when compared to column-free methods. The magnetic separators in column-based and column-free systems have a comparable magnetic force. The difference between the two systems is the column, filled with little iron spheres that pass the magnetic force onto the next over a very small distance, amplifying the magnetic force by 10,000-fold. The resulting benefit is that minimal labeling of the cells with microbeads is sufficient to retain the target cells within the column while in the separator. The iron spheres have a cell-friendly coating allowing cells to run freely through the column and can be washed thoroughly to remove cell debris and contaminants. All of this contributes to the fact that MACS columns allow for cell separation with minimal effects on cells. Combined, these advantages make MACS columns the best choice for cell separation for neuroimmunology. Well, why do we need automation? Automation reduces hands-on time and frees you to work on more important tasks that cannot be automated. It minimizes variation between different users in the lab, or even good and bad days of individual scientists, and of course minimizes costly human errors. With standard protocols and minimum user variation, you can be sure that the effects that you're seeing are due to your assay rather than artifacts. In short, automation reduces redundant experiments and allows you to focus your time where it matters. Now I'd like to introduce you to the Miltenyi Biotec automated solutions for magnetic cell separation. We'll start with the TCR Pro separator, the easy-to-use benchtop instrument for every laboratory. With fully automated sequential separation of 1 to 6 samples, the TCR provides easy access to automation without high investment costs or lots of training. You can mix many different starting materials and samples within one run. You press the run button and the instrument will label and separate the cells for you. The columns are reusable, so maintenance is easy, and there are preset programs for high recovery and high purity depending on your downstream applications. Next, we have the MultiMax Cell 24 Separator Plus. This semi-automated instrument was designed for those with higher throughput needs or for those processing lots of cells, example, from a leukopak. Finally, we have the MultiMax X. Launched in 2017, this is the instrument for the demanding high-throughput cell processing lab. Samples are labeled and isolated automatically, and you can go from an entire buffy coat to target cells in 30 minutes or from a leukopak in 60 minutes. All programs are tailored to match your needs, so we won't leave your lab until you're satisfied with the protocol and the results. In summary, the TCR Pro is great for small samples and low throughput with full automation. For example, when processing PBMC aliquots, mouse samples, or patient-derived xenografts. Because the Autoimmunity can process whole blood without phycol, it's also great for patient samples or small donor samples. On the other hand, the MultiMACS family are the high throughput variants for fast parallel processing, for example, of small human or mouse samples, but also for large blood products such as leukopacs. After you've isolated your cells, many applications require subsequent cell culturing experiments and phenotypic analysis. The importance of using optimized reagents and protocols cannot be understated, as reproducibility, not only by the investigator, but by others, depends on this. Our cell culture and stimulation portfolio comprises a specialized and versatile range of cell culture media and reagents for the stimulation, expansion, and/or differentiation of human and mouse primary cells. These include immune cells, stem cells, neural cells, and tumor cells. Small molecules and reprogramming reagents are available for stem cell research, and we offer chemically defined specialty media for the production of recombinant proteins in eukaryotic cell lines. Selected antigens, recombinant cytokines, and cell culture media are produced according to relevant GMP guidelines, enabling a seamless transition into the clinic. We offer a number of lineage-specific media solutions that are xeno and serum-free that allow you to avoid the disadvantages associated with animal-derived components that can cause significant variation in your cell cultures. These include stem cell media for iPS, HSC, or MSCs, immunological media for T cells, NK cells, and DCs, as well as neurospecific media for neuronal cultures. From the expansion and differentiation media, our medias are designed and quality tested for reliability and efficiency. Whether you're performing basic or translational research, Miltenyi Biotec develops specialized cell culture media so that you can get a head start in your experiment. Media alone, though, isn't enough to expand your target cells of interest, with many relying on the addition of cytokines and growth factors for the expansion and differentiation of your cells. So let's talk about cytokines a little bit. Our recombinant cytokines and growth factors are available in 3 quality grades: research, premium, and GMP. Using max premium grade cytokines, you can rely on the lot-specific cytokine activity value provided in the certificate of analysis. Learn how unit-specific cytokine dosing can help save you time, increase cell culture reproducibility, and reduce costs. This also facilitates a seamless transition into the clinical research of using max GMP cytokines. We also offer a number of stimulation and differentiation reagents for T cells, NK cells, B cells, MODC differentiation, and stem cell differentiation, among others. A majority of our cell culture reagents have GMP counterparts for those of you that are looking to transition into the clinic by allowing us to support you from bench to bedside. Though in order to successfully achieve this transition, you must have a reproducible product manufacturing and defined characterization methods. One common method is flow cytometry, which uses fluorescently labeled antibodies to detect both surface and intracellular proteins. Proper phenotyping is therefore dependent on the antibody and what it recognizes. So we've all heard the phrase reproducibility crisis, but how many of us really, in fact, think that we're in the midst of a reproducibility crisis? A survey carried out a few years ago by Nature showed that 90% of researchers agreed that there is either a significant or slight problem with reproducibility in research, and that this is in fact a very real problem. Irreproducible preclinical research is a global, expensive, and well-recognized problem that contributes to delays and increased costs of drug discovery. Excluding scientific misconduct, which is not a major source of irreproducibility, lack of reproducibility typically results from the cumulative errors and flaws in one or more of the following non-discrete categories of the research and publication process: biological reagents and reference materials, study design, laboratory protocols, and data analysis and reporting. Although each contributes to a systematic problem that requires extensive changes in the overall scientific culture, taking immediate steps to address biological reagent issues, specifically those for cell culture and flow cytometry, is a relatively straightforward fix that will improve the credibility, reproducibility, and translation of preclinical research. In 2011, a Nature Scientific report announced that there was a 10-fold increase in the number of retractions over the last decade, while the amount of literature had only expanded by 44%. virtually all of these came from the sciences, with biology and medicine accounting for nearly half of those retractions. In 2012, Glenn Begley, who ran the oncology division at Amgen, decided to try and reproduce 53 foundational papers in oncology. He was unable to reproduce 47 of them, which is 89%. Bayer, another pharmaceutical company, reported that in the same year it was unable to reproduce 65 of the papers in its sample biomedical literature. In 2015, various articles were published in Nature highlighting the importance of antibody validation and also called for a new generation of antibodies, recombinant antibodies that are defined by their DNA sequence. In 2016, Amgen announced in a press release that it would publish 3 studies that failed to confirm findings. These negative results will be published in F1000's newly launched published platform called the Preclinical Reproducibility and Robustness. None of these papers from the 2012 example are related to the 3 studies that they plan to publish. One published study conservatively estimates that the total prevalence of irreproducible preclinical research is to exceed 50%, with a cost of $28 billion a year in the United States alone, while other researchers have estimated that an astounding 85% of biomedical research is wasted as a result of correctable problems, tools, and resources. Irreproducible preclinical research presents a significant challenge that increases the cost and time to translate research into life-saving therapies. Despite increasing pressure within the academic community to address challenges of irreproducibility, little has been done to provide researchers with the framework necessary to standardize research. This led to the establishment of the Global Biological Standards Institute, which is a nonprofit organization dedicated to enhancing the quality of biomedical research by advocating best practices and standards to accelerate the translation of breakthroughs into therapies. This institute was established by the head of ATCC, as cell culture lines fall into the group of biological materials and are under as much scrutiny as antibodies. There are a number of initiatives that are going on with the GBSI which speak to increasing reproducibility. One is Reproducibility 2020, which is determining and providing guidelines for cell culture and antibody validation, working to standardize shared protocols and data, and providing training for PIs around reproducibility efforts. More specifically, the Antibody Validation Initiative is determining the best practices for antibody validation across the scientific community. Miltenyi Biotec is a member of GBSI and the Antibody Validation Initiative, and we have developed a new recombinant portfolio of antibodies in response. Our REAfinity antibodies are recombinant antibodies that provide superior lot-to-lot consistency and purity compared to mouse or rat hybridoma-derived monoclonal antibodies. They have been recombinantly engineered to produce highly specific antibodies that require no FCR blocking step. Additionally, they all have the same IgG1 isotype, requiring less isotype controls. The manufacturing process of REAfinity recombinant antibodies ensures high performance and consistency based on the following aspects. 1, they are derived from a defined DNA sequence, which encodes only one type of heavy and light chain, ensuring high antibody purity. 2, production is biologically and chemically defined, with standardized culture conditions in mammalian cell lines, which results in high lot-to-lot consistency. And 3, we do analytical, biochemical, and cell-based testing of every manufactured lot. The importance of the mutated Fc region is demonstrated in these 2 figures. For splenocytes, the difference between using FCR blocking or not with either REAfinity or hybridoma antibodies is not really significant. But in tumor materials, where an upregulation of the FCR receptor has been identified, our REA antibodies demonstrate a very clear advantage. When dealing with neural, tumor, and immune cells all mixed together in your starting material, it is essential that you make sure that what you are seeing is real and not an artifact of differential FCR expression that can muddy your results. So next, we're going to move on to single cell analysis, where sample clearance is a crucial step that removes unwanted cell aggregates as well as dead and dying cells. This is critical as dead and dying cells can produce toxic compounds that can influence your cell culture, as well as being susceptible to contamination, and most importantly, can have a significant impact on your downstream application. There is no better example than when preparing samples for single cell sequencing and proteomics. In a study designed with 10x genomics, we looked at the performance, reproducibility, and heterogeneity of tumor populations that were prepared using our dissociation and clearance reagents compared to those that only used our dissociator alone. The findings were that when you filter out red blood cells and dying cells and remove debris, the readout was considerably better for all three. Another common use for single-cell sequencing is to identify clones of T cells or B cells via their T or B cell surface receptors. Single-cell receptor sequencing can be very difficult and expensive. Here we show how isolating T cells prior to single-cell sequencing can allow for better sensitivity and detection of rare clonotypes. The single-cell TCR sequencing was performed to assess clonality of tumor-infiltrating leukocytes in this example, and we were looking at either bulk or isolated T cells. The target number of cells for sequencing was 10,000 per sample, and we here list the top 50 TCR beta CDR3 clonotypes. And what you can see is that the isolated T cells, we were able to pick up on a number of clonotypes that were not detected using bulk cells. An additional tool to single-cell sequencing, single-cell proteomics is now becoming an essential tool to identify effector functions of various cell types. Isoplexis is a company that offers single-cell proteomic solutions, and they have established protocols that rely heavily on the magnetic cell separation of samples using MACS technology. In this example, they focus on various checkpoint and combination therapies for T cells, TILs, and PBMCs in the context of various cancer models. The quality of the data that can be acquired using single-cell proteomics and sequencing is dependent on the quality of your starting materials. So make sure that your time and money are spent wisely when it comes to sample preparation and cell separation, as these costs are dwarfed by those of the downstream applications. The last component of our neuroimmunology workflow is our new imaging platform that allows for the visualization of whole tissues, including entire mouse brains. The ultramicroscope is a light sheet microscope that was designed for imaging large, cleared samples. Light sheet microscopy is based on the 90-degree principle. That is, there's a separate light path for illumination oriented orthogonally to the imaging light path. This provides for illumination of a selective plane or sheet within a sample, which eliminates out-of-focus illumination or emission and enables 3D imaging and high-speed camera-based image acquisition. There are 4 important characteristics to be mentioned about these systems. It is very user-friendly and easy to operate. Its design allows the easiest exchange of samples, imaging from overview to region of interest at a higher magnification only by turning a knob. It has a unique light sheet illumination which guarantees superior image quality even in larger fields of view. Third, the system can handle all current clearing solutions. And finally, the ultramicroscope is a system for imaging large samples. Data using the ultramicroscope, too, has been in the spotlight recently, with numerous covers of Cell, Nature, and the Journal of Neuroscience featuring images acquired with this technology. So this wraps up our presentation of the Miltenyi Biotec workflow for neuroimmunology. So let's take a quick look at some key applications and recent publications that focus on various neuroimmunological diseases. As mentioned earlier in the presentation, there are 4 primary applications of neuroimmunology, including cancer, autoimmunity, degenerative diseases, and other. Let's start with cancer. Brain cancer is a relatively rare yet very deadly disease. Tumors can form anywhere in the brain. A majority of tumors form in the frontal, parietal, or temporal lobes, but as mentioned, nearly any region of the brain can develop tumors, as shown by this figure. Of the brain cancers that are malignant, 80% of them are gliomas. Glioblastoma makes up a vast majority of the deaths in brain cancer and is the primary cause of death in children with brain cancer. Despite significant research efforts, the mortality rate of glioblastomas has been static for the last 30 years. This graph shows that how the advanced stages of glioblastoma have extremely low survival rates and have prompted researchers to look at new ways to treat this disease. One of the new approaches is to use immunotherapies. In this example publication focusing on glioblastoma, the researchers looked at tumor-associated macrophages and CD8 T cells during combination checkpoint therapies. This group utilized our gentleMACS octo with heaters, our brain and tumor dissociation kits, the myelin removal microbead kits, and utilized the MultiMAX 24 to isolate CD11b positive cells using our CD11b microbeads. There are other example publications in autoimmunity dealing with multiple sclerosis, utilizing our microbeads and cytokines. We have papers on SLE and encephalitis utilizing our cell separation technologies, and we also have it on viral encephalitis. If you look at some of the various degenerative diseases, you can find recent reports dealing with Alzheimer's, Parkinson's, as well as ALS. And finally, there are reference papers that identify immune components and utilize MAX technology dealing with stroke and traumatic spinal injury, various infections, including Zika, and various psychological conditions that have immunological components. We have gone through the entire neuroimmunology workflow, starting with sample preparation to obtain a single-cell suspension from a solid tissue, cleared of debris and dying cells for subsequent cell separation using MAX technology. We highlighted the importance of having reliable reagents for reproducible cell culture and phenotypic analysis, including the reproducibility crisis associated with antibodies and cell lines. We talked about the importance of having quality starting materials for single-cell sequencing and proteomics, and finally introduced you to the newest edition of our workflow, the Ultramicroscope 2 for the 3D imaging of whole tissues. Thank you for your time, and please stop by the booth for additional information about the offerings from Miltenyi Biotec.